Hello everyone, my name is Barbara Meyersand and I'm a PhD candidate at Wageningen University in the Netherlands. This video is the first one of the Team Trace series of short videos on sample collections for genetic and chemical analysis. In this first video, our goal is to explain about the type of samples, the sampling design, and the field form. So what will you need? First of all, printed field forms, preferably printed on waterproof paper, a pencil, a GPS device, and lots of batteries, better if the batteries are rechargeable, a DBH tape, a camera or a smartphone, and scale paper. So first thing, find your tree. Once you've done that, you need to ensure that the tree is the species that you are looking for. In case the timber is produced by one single botanical species identification can be done by an experienced prospector. If that's not the case, we recommend you to be accompanied by a taxonomist or to take botanical vouchers. Our sampling strategy. So we target a minimum of 30 trees per species per site. To have samples that represent the area, we avoid to collect samples too close together, and we spread the collection area. For that, all trees must be at least 100 meters apart from each other to avoid sampling too close related trees. And for that, make sure to check in the GPS that the closest sampled tree of that species is at least 100 meters from the next tree that you are intending to sample. Something else that you need to keep in mind is that at least two trees must be at least 3 kilometers apart from each other. The 30 trees must be sampled within a radius of 5 kilometers, as shown by the green circle on the illustration. For example, in this site, we have a maximum distance of 7.3 kilometers, which is within the radius limit. Now zooming out, we can see the distances between three sites we sampled during one expedition as an example. Filling up the field form. This is our field form. You should give a unique number for the tree to be sampled, called the tree ID, that must also be written on all samples collected for that given tree. Specify the species. If you are uncertain about the species, only write the genus. It's better to have a higher level of botanical identification than the wrong one. You can always indicate in the remarks if you have a good guess of which species the tree is. If you are in a forest concession, you can write down the number that the company has given to the tree, the prospection number. It can help you to retrieve or check the information later on if needed. In this field, you indicate if the tree is still standing or logged and the logging date. Please don't collect samples from trees that have been harvested longer than 1 month, and make sure that the inner part of the bark is not with fungi, dry, or with dark spots. Then you measure the diameter at breast height, known as DBH. That means 1.3 meters from the ground, and write it in the form in centimeters. If the tree has buttresses, measure the DBH just above it. For felled trees, you can measure the stump diameter. Now it's time for the geocoordinates. Make sure that the GPS is calibrated and that the format is set to UTM coordinates. Create a new point in your GPS and name it with the tree ID, like this. That done, write down the coordinates with latitude, longitude, and elevation on the field form. Here, you can write down whether the tree is within 25 meters of a river or a stream. In the remarks column, please include information such as if the samples are collected from a stump, the local name, and odd characteristics such as bark color, if there was a hollow tree, if there are fruits, if you collected a botanical voucher or not, the type of samples that were collected for this tree, etc. As a last step, take pictures of the bark, the leaves, fruits, and flowers on a scale paper to be able to check the botanical identification later on. Make sure that the tree ID is included somewhere in the picture. Please also take additional photos of any oddities you wrote down in the remarks column, and then repeat the process for every sampled tree. So this was our first tutorial. Check the other videos in our blog, teamtrace.nl, if you want to know how to collect cambium, wood, and soil samples.